Good afternoon and welcome. My name is Pat Furlong. I'm with PPMD, and we're so really delighted that you've joined us today for this really important webinar. Dr. Eric Hoffman is with us to talk about Vamorolone, and Suzanne Gaglianone is joining him to talk about her role in the development and execution of clinical trials with Vamorolone. I think as a community, we've always been interested in the use of steroids and what they have done in terms of prolonging ambulation, and also noticing the side effects that for some are really difficult. So Eric, in the drug development process, has been working on a drug that has similar benefits of steroids, but fewer side effects. And so with that, Eric, I'm going to turn the meeting over to you.
Thanks, Pat. I'll start off with a couple slides on what steroids do as far as the benefit to Duchenne, both deflazacort and prednisone, and then talk about the side effects a bit, and then talk about the Vamorolone program and where we are in development and what trials and what we've found to date. And Suzanne will follow up after I give the science and clinical trial end with information on the trial that's currently enrolling and how we can, if anybody's interested in that, facilitate participation in that. One thing right here is all the different organizations and people that are involved in the Moreland program for about the last 10 years. So this has been supported by PPMD and many foundations. We're very grateful for that support. It's really a partnership between governments, both in the U.S. and the European Commission, and a dozen foundations, and then academic groups at University of Pittsburgh, Newcastle University, the Cooperative International Neuro and the Muscular Group, and more recently there's been some support on a option agreement from Idorsia and Centera. There have been some questions about press release with Centera recently. Just to be clear that the entire program has always and will continue to be managed by ReveraGen and the people working for ReveraGen. The press release was regarding a future option agreement for sales and marketing once the drug's approved. So it really has little impact other than providing some milestone payments that help continue the program.
Thanks for clarifying here.
Sure. So the first is what treatments have shown long-term improvements in strength and mobility. Now there's a number of treatments, even physical therapy, that certainly delay progression, but there's really only one drug or one class of drugs, which is the corticosteroids, that actually show short-term improvement as well as then stabilization, and that's it. So prednisone and deflazacort. So what do they do? Well, there's been some series of nice publications over the last year, some from the Cooperative International Neuromuscular Group, or Synergy. This is Craig McDonald's recent Lancet paper, and you can just see the blue line here is at what age do kids take longer than 10 seconds to stand up from the floor. And you see that kids without glucocorticoids lose that pretty much uniformly before 10 years of age, or lose it in the sense of taking longer than 10 seconds. But with glucocorticoids, that's extended for quite a bit. So here, about 50% of patients can still stand up from the floor in less than 10 seconds at, say, 12 or 13 years of age. And then that follows through not only with time to stand from the floor, but loss of ability to stand, loss of climbing of stairs, loss of ambulation, and also upper arm functional milestones. You see here, again, without glucocorticoids and with glucocorticoids, and again, this is age along the x-axis, you see that all those milestones are delayed with glucocorticoid use. And so in general, these different milestones, functional milestones, are improved or retained about 2 to 5 years with glucocorticoids compared to not having glucocorticoids. Now, a lot of people ask about the differences between deflazacort and prednisone, and again, we published this in the Synergy group, looked at 340 kids with Duchenne over time, and compared prednisone with different doses, some daily, some 2 days a week, and looked at deflazacort. And here again, one of these graphs, you're looking at percentage of ambulatory patients as a function of age, over long term, they seem similar, but over the in the middle age range, deflazacort actually does better, and that was seen. One of the complications of interpreting differences of prednisone and deflazacort is there's many different schedules of corticosteroids given, and you see some here, daily versus 2 days a week. Deflazacort is generally given daily. Deflazacort does seem better in the long term, but it ends up being dosed higher. And so that's explained a bit more here in the next slide. So this is a summary of a number of the papers, but particularly this one on deflazacort versus prednisone. Again, of 340 patients, you see about a 3-year delay in loss of ambulation, but there's 14 different regimens of dosing of how many times a week or what dose of steroids are given. Non-daily treatment was common for pred, but rare for deflazacort. So even these two drugs are being used differently. So whereas prednisone is often not given every day, deflazacort is almost always given every day. Deflazacort is associated with a later loss of ambulation, almost a 2 years difference. But again, there's this dose difference. Prednisone is given at lower dose than is deflazacort. And even in the study we did, deflazacort is supposed to be dosed at 0.9 mg per kg per day. That's what's recommended. But you see in practice, people are using less. With that, deflazacort showed higher frequencies of side effects. So clear differences, more growth delay with deflazacort, more cushingoid appearance, more cataracts. The only side effect that didn't seem to be worse with deflazacort was weight gain, where deflazacort showed actually a little better weight gain. So in summary, the use of deflazacort was associated with later loss of ambulation, so better improvement, but also that came at a cost of increased frequency of side effects. So it could a lot could be dosing, that just because deflazacort are used at higher doses, you might get better benefit, but you get more side effects as well. What kind of side effects are patients concerned with? Well, there's lots of side effects of glucocorticoids, and one that parents actually rank as number one, I think a lot of clinicians, is what's called bone morbidities or bone fragility. And here is a recent study out of Canada, looking at 49 DMD boys on long-term deflazacort, and what you see is spinal fractures caused by the drug. So 1 in 5 kids will show a vertebral fracture, a spine fracture by their 3rd year of therapy, 50% by 5 years, 70% by 7 years, and 100% with 9 years. So there's one of the key side effects. There's a high risk of spine fractures associated with the length of deflazacort treatment, regardless of age of start of therapy. So recently, with the help of PPMD and others and MDA, we sent out a survey and asked parents. So this is about 91 parents responded to the survey recently. And what do parents view that where their Duchenne children are taking glucocorticoids? What's of most concern to them? And number one was loss of bone density, where the majority of parents, almost 80%, were very concerned regarding loss of bone density. Weight was also up there. Suppressed immunity was also up there. Mood and changes in behavior and stunting of growth were also about more than 40% of parents were very concerned. So with all these different I should point out that some weren't so concerned. This is a wrong graph. I should have fixed it. But cataracts, sleep disturbances, skin changes, even cushingoid was of less concern to many parents. Okay, so corticosteroids clearly improve DMD kids' strength, but they add quite a few additional problems as well, and that's where we come in, as Pat said. We set out about 10 years ago to design a drug that retains or improves efficacy or benefit, but reduces or eliminates side effects or so-called safety concerns. So how did we do that, and how far have we come? Well, the first thing is we wanted to design a better steroid, and we have to understand how steroids work to design a better steroid. And despite these steroids being one of the top drugs prescribed in the world, I think they're number 2 or 3 since the 1940s, how steroids work has really become clear just in the last year or two. We've always known, and this is key to everything I'll present, and its key 2 words are activation and repression. So the way to think of this in all these subsequent slides is activation seems to activate side effects. And that's a receptor drug. So these drugs bind to what's called the glucocorticoid receptor. It's a protein in all your cells. And when they do this transactivation activity, it seems to not so much be benefit, but mostly side effects. On the other hand, transrepression, think of repression as repressing the inflammation in muscle or repressing inflammation. That seems to be most of the benefits or the efficacy of the drug. So if you can separate transactivation, start shutting down transactivation, and keeping or improving transrepression, then you should have a better steroid. And so that's what's summarized here. Retain or improve transrepression, the anti-inflammation, but lose or reduce transactivation, which is the activation of side effects. So it's just, there's a lab at Emory, Eric Ortland and his lab have really made a lot of progress in defining at the molecular level what the differences of transactivation and transrepression are. And so what you see here is the same receptor. This is the protein that binds the drug and then goes into the nucleus and interacts with DNA. Here's your DNA helix. And you see when it's bound to the drug, it hits DNA at 2 points. This is called a dimer. It's two different receptors paired up, each with their drug. And it touches the DNA at specific sequences. And this transactivation requires what's called coactivators. These are additional proteins that bind and stabilize that interaction. And again, that seems to be mediating those side effects. What's become clear over the last just year is transrepression is a completely different thing. You still have a receptor here, 2 of them, but only one side of it binds the DNA, and it binds a completely different sequence than transactivation. And so here are a bunch of inflammatory genes, and you see that the receptor is shutting them down by binding just this small sequence here. And you need a whole different set of proteins called co-repressors to stabilize this interaction. So now we know really what's the difference between transactivation and transrepression. Transactivation is 2 copies of the receptor sort of married, each binding to DNA through these sequences, and they have coactivators. Transrepression binds only one side, a completely different sequence, and it needs different proteins called co-repressors. So in summary then, the molecular action of glucocorticoids, the drug or the ligand and the receptor, goes into the nucleus. Transactivation is this interactions with this 2 sequences, and that's mostly side effects. Transrepression goes and blocks gene activation, that's anti-inflammation, and this requires coactivators, and transrepression requires co-repressors, and they're key players. So this is where we come to Vamorolone then. And so what we did is John McCall was a chemist. He designed a kink in the steroid. So this blue is prednisone, and you can see the structure of prednisone and deflazacort here. And the key thing he changed was where this blue arrow is. That oxygen there interacts with the receptor, and we took that out and added a double bond. And that causes this kink here. And that kink starts changing how the drug interacts with the receptor. And so what we've recently discovered, along with Eric Ortland's lab, is here you see the receptor, again, the drug in the pocket there, and this is Vamorolone binding to this receptor. And here's one of these co-repressors. Vamorolone changes its interaction with the protein receptor, pushes this area out, and changes the interaction with these coactivators and co-repressors. So remember, coactivators are side effects. Vamorolone binds 3 times less than prednisone. Co-repressors are efficacy. Vamorolone binds 3 times more than prednisone. So it's through these different interactions that we're separating transactivation and transrepression. That's how it becomes a dissociative steroid. Okay, so how does it work? Well, it works pretty much as would expect or hope. Here's an assay for transactivation. You see prednisone turning it on, Vamorolone doesn't. Here's another assay similar. Here's transrepression efficacy. You see they work almost identically. So it seems, again, we're separating side effects from efficacy. Now, while we were developing this, John McCall's original idea was maybe Vamorolone would be a good membrane stabilizer that would help counteract dystrophin deficiency. And what's become clear is that's true. There's a nice paper from Jody Jaiswal's lab at Children's National, just came out a couple months ago, where in fact Vamorolone does counteract membrane instability. He showed it in dysferlin deficiency, but the same thing's expected to happen with dystrophin. And prednisone actually makes it worse. So, again, a different activity. And then Chris Heyer just had a paper, this should be 2019, it was just accepted a week or 2 ago, where it's called the mineralocorticoid receptor, which is what eplerenone and spironolactone are given to Duchenne kids to help with their heart. Those are called mineralocorticoid receptor antagonists. Vamorolone is just like eplerenone or spironolactone, and it helps the heart. And that's opposite of what prednisone does. So prednisone actually is an agonist instead of antagonist. So I'll just show you just a little bit of data for that. Here's the heart improvement showing that we've really completely changed how the drug acts with this heart drug. We've changed what prednisone is an agonist to instead vamorolone is an antagonist. And you can see here a large heart is an unhealthy heart, and you give vamorolone and it turns it back to normal. And again, that was just recently published. And here's Jody Jaiswal's paper showing membrane stability, looking at vamorolone and prednisone actually makes membrane stability worse, whereas vamorolone actually improves it towards wild type. So it's a membrane stabilizer as well. So in summary, vamorolone is a first-in-class steroid drug. I should say that vamorolone is not a glucocorticoid. It has structural differences, so it is not in the same class as prednisone and deflazacort. Changes interaction with the receptor with structural effects on this co-activated co-repressor domain. It binds co-repressors more, which is good, co-activators less, which expects less side effects. So we've lost this transactivation with retained transrepression. We've changed this mineralocorticoid heart drug from an agonism, which is bad, to antagonist, which is good. So we've almost added a new type of drug on top of it, activity, which is eplerenone. We've gained membrane stabilization. And in preclinical models, like mouse models of Duchenne, we retain or improve efficacy, and we lose most of the safety concerns. Okay,
so Eric,
let's
can I just ask you a real quick question there?
Sure.
The studies that you've just presented are on prednisone and not deflazacort. Would you expect that based on the molecules deflazacort and prednisone being similar, that the effect on the membrane stability as well as the heart would be the same, prednisone and deflazacort, or that deflazacort may have different properties based on some molecular change in its development?
Yeah, deflazacort and prednisone are really quite similar. Deflazacort was first developed to change its mineralocorticoid receptor activity. So as I mentioned, prednisone binds. It's not a heart-healthy drug, so it's not like eplerenone. It does the opposite. Deflazacort was developed to be agnostic to the mineralocorticoid receptor. It really doesn't bind it at all. So it's not good or bad. It's just neutral. Whereas vamorolone binds it in a good way. So we expect the deflazacort and prednisone to pretty much be identical across all these different activities except the mineralocorticoid receptor, where prednisone could be viewed as bad, not good, deflazacort neutral, and vamorolone good.
Great. Thanks very much.
Sure. Okay, the last slides I'll present before handing it over to Suzanne is on the clinical findings to date. So when you do clinical trials, you do what's called first in human, which is the first time you try the drug in a human, and that's often in volunteers, so adult volunteers that join the trial. And we studied a very large dose range from about a tenth of prednisone dose to 30 times prednisone dose, and looked at 86 volunteers, and we saw what's called no dose-limiting toxicities. In other words, we could have technically kept on giving more and more every day because we weren't seeing any problems with that. We did biomarker studies with that, and this is published, where we saw loss of bone problems, loss of insulin resistance, metabolic, adrenal suppression was much less, and we didn't see any immune suppression. So already, but this was just 2 weeks of treatment in adult volunteers, we saw that we were losing many of the side effects of glucocorticoids. So then we went on to a Duchenne study. This was 48 Duchenne boys, ages 4 to 7 years. The dose range was, again, broad, but less broad than in the adult volunteers. We went from about a third prednisone dose to about 9-fold or 8-fold prednisone. We found one of the key things is you look how the body reacts to the drug and how it metabolizes it. So that's called pharmacokinetics. and we saw it was really similar to prednisone, where we're just giving a pulse of drug in the morning and it's pretty much gone the rest of the day, and that's the same thing that happens with prednisone and deflazacort. Again, didn't see any dose-limiting toxicities. Again, could have presumably gone higher. And again, we did these biomarker studies, and these are recently published, where, again, the bone morbidities, all the biomarkers looking in the blood seemed to pretty much go away. We didn't see the bone problems, much less insulin resistance, adrenal suppression, and immune suppression problems. Now, we also had biomarkers for efficacy. So these were pro-inflammatory proteins that we'd studied in Duchenne and a couple other diseases. And we would always see them go down with glucocorticoids. And these were these inflammatory markers. And so we could see a nice dose response to this as well. And I'll show this on the next slide. But we also saw a decrease in CK. Remember, we thought that this would be a membrane stabilizer. And in fact, after 2 weeks treatment, and I'll show this on the next slide too, we saw a decrease in creatine kinase, suggesting that it was having a membrane stabilizing effect.
So, Eric, why did you stop at 6 mg when you could have gone up higher? What was the rationale to get a 6 mg dose?
You know, that's a really important but tough question. It gets into how even prednisone is dosed in deflazacort. And you can see that the dosing schedules, as I mentioned earlier, are sort of all over the place, particularly for prednisone. And it's really not clear what a good dosing regimen is for prednisone in many diseases. It seems to always be this balance of side effects and efficacy. And with Vamorolone, if we're losing many of the side effects, does that change the whole schedule? Should we be dosing twice a day or 3 times a day? And it's really hard to say. We have to start somewhere, and so we thought that 1/3 to 10 times prednisone dose was a good way to start. But in the future, there may be studies of Vamorolone twice a day or at lower or higher doses. But you can see that just the first studies, you have to start somewhere, and it is a judgment call.
Yeah, I don't disagree, but we just had that inflammatory meeting, and inflammation plays such a key role, especially in early years when it then moves into the fibrotic area after, age 10 or plus or minus, depending on the child. But it would seem that if you could go up in this and really dampen that inflammatory response, that might be really useful. Just a thought.
Yeah, and I'll show in this next slide what the biomarkers look like with regards to that dose response. Here's just the background on these trials. The first studies in 48 kids were actually a sequence of 3 trials. This is phase 2A was 2 week on drug, 2 week off drug. 2A extension was 6 months at the same dose for 6 months longer. And then the long-term extension is 2 years, where all the patients on low doses can have escalated to high doses of Vamorolone. So now, currently, we have kids that have been on Vamorolone as long as 2.5 years, and all of them are on high dose, either 2 or 6 mg. So it's about 3 to 9 times prednisone dose. These are the people involved. Synergy site, the Synergy group, same that did the natural history study, was the one who did this study. So here's just biomarkers, and this data is published again just recently off the 2 weeks treatment. And here you see the lowest doses and CK after 2 weeks of treatment and 2 weeks off treatment. And you see that CK comes down then by the 2 higher doses. The highest dose, it comes down at 2 weeks, and then it stays down even after stopping the drug for 2 weeks. So it does seem to have a membrane stabilizing effect. And here's some of those inflammatory proteins in blood. So you see they're high at the lowest dose, and they don't change. But as you increase the dose of drug, they go lower and lower. And Pat, we've benchmarked these against pediatric inflammatory bowel disease patients recently that also get steroids. And these decreases at the highest dose are similar to prednisone in pediatric inflammatory bowel disease that also treats the disease and brings these biomarkers down. So at least the highest dose is similar to what's efficacious in other disorders.
Great, thanks.
So the key thing is you guys obviously want to know how the kids do. And so what we see is improvement. Again, this is not stabilization of the disease. This is quite significant improvement over 6 months of treatment. So all these graphs have the 4 dose groups. So this is the lowest dose group. the next dose group, the next dose group, and the highest dose group. And you see the two high dose groups actually improve their speed of standing up from the floor, and that's statistically significant. Now, we don't have a placebo. This is what's called an open label, and it has to be open label because we first treat all the kids at low dose, make sure they're okay, before we recruit the next kids at the next dose, make sure they're okay, and that's what's called a standard dose escalation study, which is open label. But we can look at like this. Sorry?
It was also mutation agnostic, right?
Right. This is all Duchenne kids. There is no mutation requirement other than just having Duchenne and being steroid naive. So we can put the Synergy Natural History Study in there. Now, this was done quite a while ago, 10, 15 years ago. So it's not exactly the same, but it sort of sits in there. And again, you don't see this improvement over time, and that's also statistically significant versus Vamorolone. So let's look at a couple others. This is time to run walk. This is your 10-meter run walk. Again, you see the highest dose group shows a dramatic improvement. The next dose group initially improved, and it seems to go down, but again, these are small studies, open label, so it's hard to say whether that's significant or not. If we go to, and then I also showed in this slide the Synergy Natural History Study. So here's the Synergy Natural History Study. Again, a slight decline or stable. So then the 6-minute walk test, the highest dose group had an average of 45-meter improvement in the 6-minute walk. And again, clear dose effect, lowest dose group, next dose group, next dose, and next group. So all these low dose groups have now escalated and been on a year of treatment at the high doses. And so we'll get that data soon. There's North Star Ambulatory Assessment. I'm sure many people are familiar with that. It's about 17 different tests that get combined into a score. This has a big developmental component because it's how the kids are developing over this 4 to 7 year age range. So you can see even the low dose kids are improving just because they're getting older and improving in their skills. There does seem to be much more improvement, but this is not statistically significant. In terms of side effects, adverse events and serious adverse events, again, we just didn't see much. So there was safe to highest dose tested 20 in adult volunteers. The 2 weeks was safe to highest dose tested. The 2A 6 months was safe to the highest dose tested, and this 2 years is ongoing. But there have been what are called no dose-limiting safety concerns in either adult volunteers or DMD. Now, the next type of safety is the glucocorticoid-induced side effects. So we call this pharmacodynamic safety. This is a bit different than just straight drug toxicity. This is actually side effects induced by the prednisone, the glucocorticoids, and the deflazacort. So a key one, again, is bone. We're really concerned about that. And we didn't see any changes in bone biomarkers in the adult volunteers to very high doses. But it was only 2 weeks of treatment. Insulin resistance, that's the metabolic changes, why they're called glucocorticoids because of glucose effects, no changes. Adrenal suppression we start seeing at high doses, much higher than you see. You see adrenal suppression at much lower doses of deflazacort prednisone. Now the 2 weeks treatment was similar. Bone biomarkers, no changes in bone biomarkers, insulin resistance. At the highest dose we see a bit of increased insulin, but measures of actual insulin resistance, HbA1c, were normal. And again, we see at high doses more adrenal suppression. So what are the side effects of greatest concern that are reduced with Vamorolone? So we compared our survey of parents to other surveys of steroid-treated adults and also a Henriksen study of DMD parents. And again, across you see loss of bone density is way up there. Weight gain is high up there. Now, with weight gain, the highest dose of Vamorolone shows similar variable weight gain to prednisone. So while we're about 8 or 9 times the dose of prednisone, at the highest dose, some kids show weight gain. So that's the one side effect we seem to still have retained, but only at high dose. The 2 mg per kg doses doesn't seem to have that, but a subset as the highest dose for Moreland seems similar to prednisone. Stunting of growth, we don't expect that because we're not seeing bone biomarker changes. And as far as mood and behavior changes, so far there hasn't been any reports of that. So it seems to be okay as far as that, but it's hard to measure that. Okay, where do we stand? And then I'll go to the last the trial that's currently enrolling. As I mentioned, after this original 48 patients, 43 of those are now heading towards 2.5 years of treatment. And remember, they're not going to standard of care. So they're not doing prednisone or prednisone. And it's the physicians and the parents that are deciding that. They can drop from this long-term extension at any point, but there seems to be a lot of satisfaction of both the patients and the physicians with Vamorolone. I think that's very encouraging. And families and physicians in general, there's only been 2 patients that dropped because of weight gain and 2 others for other reasons unrelated to the drug. So that's where we are currently. All those patients are finishing a year of treatment within the next couple of months, and then 2.5 years of treatment by the April 2020. And now what we've started enrolling in is a new study called 004. It's 4 to 7 years, steroid naive, 120 boys. And what we've also started to work on is a safety study of 2 to 4-year-olds and 7 to 18-year-olds. And the current design allows previous steroid treatment. So would wean off deflazacort or prednisone and then transition to Vamorolone. And we hope.
And how long is the wean-off period?
2 to 4 weeks. Well, actually, you know what, Pat? Disclaimer. This study is still under design, so we haven't decided anything yet. We've just started it, but the current, I don't even know if I should say anything because it's so preliminary. But, we're thinking maybe we don't need a wean-off. Maybe we can just transition straight. So that's what we're currently discussing. And we're including parents in these discussions.
Yeah, because it would be hard to, I think the washing out of these medicines, especially what none of us want to find out is how much improvement we were seeing that we didn't recognize in the washout period. Yeah. That's
exactly what we're thinking as well. Yeah.
And the boys on the extension?
Sorry?
And the boys on the extension are able to continue, right?
Right. Yeah, what we've done is Lori Conklin's taken the lead on extended access plan, and that's submitted to the FDA, and so would permit all patients on Vamorolone to continue indefinitely, if they wish.
Thank you.
Sure. And it's different. Those extended access plans are different for every country, so we're working through each country, but it's complicated.
Yeah.
I'm sure
it is complicated. But I appreciate the idea or the concept of thinking of no washout period from a steroid, because I think that's, as I say, no one wants to find out at the end of the day that they're going to lose strength that is unlikely to be recovered.
Right. Okay, a quick description of this phase 2B study. The study chairs are Michaela Guglieri at Newcastle and Paul Clemens at University of Pittsburgh, and again coordinated by the Synergy Group and the TRIMS at Newcastle University. The design is 2 periods, each about 24 weeks or 6 months. The first period is randomized to 4 different groups. 1 of 2 doses of Vamorolone, 25% to placebo, 1 in 4, and 1 in 4 to prednisone. But that's just for the first 24 weeks, and it's all blinded. After that 6 months, the placebo and the prednisone cross over to Vamorolone. So then everybody in the second 6 months gets Vamorolone, and once you finish this, you can go to the extended access plan. So again, long-term access to Vamorolone.
And what's the dose of the pred?
0.75.
Daily?
Yep. Which is interesting because, as I just pointed out, Pat, that, in practice, kids are dosed less than that.
Exactly. Yeah.
But that's the recommended dose. This has been designed with parent involvement, burden kept to a minimum. We try to keep it at about 1 visit per month. The dosing is just by mouth. It's just an oral solution. Now, in this case, because we have both placebo and prednisone. Prednisone is a pill. So this is what's called a double dummy design. Each kid takes 2 things, a liquid solution and a pill in the morning. And so the liquid solution will be either Vamorolone or placebo, and the pill will be prednisone or placebo. But then everybody switches over to just liquid Vamorolone in the second 6 months. And here's the, so all dosing's done at home, just in the morning. There's no, you don't have to go to the hospital or anything for that. And these are the North American sites, and the blue ones are currently active. The others should be active shortly, which is in the next couple weeks. On the blue, the blue ones were the ones that participated in the phase 2A and are also taking part in the phase 2B. And these are the United Kingdom, Israel, Australia, Sweden, Netherlands, Czech Republic, and Belgium. And we're just in the process of probably adding Spain and Sweden, or Spain and Greece as well. And again, these blue sites were previously in the previous trials, and they're still following the kids in the LTE. Okay, I'm handing it over to Suzanne. Suzanne, you want to introduce yourself as far as your history? I guess the slide goes on. And then Suzanne works for ReveraGen and actually was suggested by Parent Project and Pat, and it's been wonderful to work with as a parent who has a lot of experience with clinical trials with her own son. So she coordinates all the patient travel for ReveraGen.
Yeah, Suzanne, we're really happy to have you. One real quick question for you, and this is maybe a silly question, Eric. Have you tasted it? And is it awful? Because as you know, some of the kids have sensitivity to different tastes. So I'm wondering how much of Vamorolone have you taken, and what does it taste like? And then we're delighted to have Suzanne.
I personally haven't, but I have heard other people working for ReveraGen have. And the kids seem okay with it. It's a citrus flavored suspension that's slightly artificially sweetened. It really has no taste.
Right.
The drug itself.
Okay, I appreciate that. Suzanne, I've known Suzanne since Michael was diagnosed, and I'm really thrilled that she's working with ReveraGen. She is really a wonderful person, and it's up to you now, Suzanne.
Thanks, Pat. Thanks, Eric. So good afternoon, everybody. So this title of the slide is I've been in your shoes. So as Pat mentioned, my son Michael has Duchenne. He's 23 years old. And over the years, we have participated in many different studies. There might even be more on this. I'm not even sure. We tried to come up with what we could remember. But we understand the importance of participating for drug development, but we also definitely understand the difficulties that sometimes occur when people have to participate and leave work and travel and such things. But not only do I recognize it, but ReveraGen did when they designed these studies, and Eric touched on it. But they got feedback from the Duchenne community, looked at what they could do to design the trial to be least burdensome, least amount of visits as possible, least amount of testing as possible, but still gain, the data that they need to get. So my job is patient travel coordinator, and part of that is support for families. So for travel and stay support, we try to eliminate any as many out-of-pocket expenses as we can. So prepay hotels, prepay airfare, if maybe there's parking I can prepay, whatever I can do to eliminate the out-of-pocket expenses, we try to do. And then there's access to one person, and me, for any questions or problems. And it doesn't have to be travel related. It could be, anything that comes up. At least there's one person that they know they can contact. If I can't help, I'll find somebody on the team that can help. And hopefully that, helps ease some of the stress that can happen when you're participating. Another goal was to make access to sites as easy as possible. So for the 2B study, there are 30 different sites, 9 different countries. We try to have them distributed well geographically. In fact, we just added 2 more sites in the States because when we looked at the map, we realized there were some gaps. So, try to focus on patient needs, when we can. So I'll just touch on the process of how it works for us, and we hope that the patients find that it's an easy process. So once a patient is identified, screening is scheduled. The site provides the parent with our guideline, our reimbursement guideline, and that details what we pay for, how we pay for it. And then I work typically with just the parents, sometimes with site coordinators as well, but just to schedule out the dates, look to see when they're going to need hotels, if they need airfare, whatever I can do to make that stay a little bit easier. For additional expenses like food, transportation costs, anything that's not prepaid, receipts are submitted to me, and then we reimburse as needed. So in this whole process, we try to be flexible. We try to have compassion. Everybody understands the situation that the parents are in. So I'll give you two examples that I thought were really great and made me very happy to help. So there was an international patient who very much wanted to participate, but they were approaching age 7 when 2A was starting. The site in their country wasn't going to open when they thought it was, and the child was going to age out. So there was a site in the States that was willing to take the patient, and ReveraGen actually paid for the family to come to the States. They stayed for the first month to reduce the burden of going back and forth. And they've continued to participate in the trial and travel back and forth. And it's been great to work with them. And in the flexibility also, on our guideline, it says that one parent or guardian can travel with the patient. And that's what we will pay for. But there's a participant who has special needs beyond Duchenne. And it was just going to be just too hard for either parent to travel alone with the child. So we made an exception, and since the beginning, 2 people can travel with the child, and we certainly hope that makes that much easier for them to participate.
Thank you, Suzanne. It's amazing to have one person. It's amazing to see your flexibility. So thank you all for that.
Yeah, it is. I think it really does help. I think Eric pretty much did this next slide. I think parents are always wanting to know about when we will get approvals. I know everybody wants that. So we will be looking at applying for approvals with FDA and EMA sometime between the second and third quarter of 2020. And the timing of that is the end of the long-term extension, and then hopefully the data from the blinded portion of the 2B, so the first 6 months data, and we're hoping to do that then. Some questions that have come up. So when will trials of older and younger DMD patients be carried out? So Eric mentioned at the end of this year, we're going to look at a safety study for broadening our look at the who can take the drug. And then also the EMA has approved a pediatric investigation plan that's going to look at a very large range from newborn to 18 years. I think we talked about the 2 to 6 mg per kg. I just want to point out that 2 things. One, that some thought that the 6 mg was a lot, 3 times what the other dose was, but and oftentimes the regulators want to know why we don't go higher. So the 6 mg, we thought was, or they thought, people at ReveraGen who know about this thought that this was a good design. And also, interestingly, in the long-term extension and the EAP, we do allow for an intermediate dose. So there's a possibility to go to maybe 4 mg per kg. And in the end, I think we all know that everybody's an individual, reacts to drugs differently, and so optimizing a dose for individuals can be done. So if I join the Vamorolone trial, will I be able to join gene therapy trials? So patients can stop Vamorolone and transition to deflazacort and prednisone, and I think probably that's what people have on their minds. Can we do that? And yes, people can do that. The other thing about bone breakage, I think, and as a parent, I know I worry about the bone density from being on deflazacort for so long. But those bone turnover biomarkers that Eric mentioned before are not seen with Vamorolone. So I think that that's a good sign. And then we're going to look at spine fractures. Now that we have a lot more data over time, we can look at that. And finally, just thank you to all of our partners who make this possible. And we can open up to questions.
Yeah, thank you, Suzanne. That was really good. I'm going to start off with a couple questions on if we sort of go back to that extension study. So the folks that have been in the extended access program following the phase 2A, I think that's right. Will their burden in terms of being seen at the clinic, et cetera, and travel related to that extended access program, will that be less or changed along the way as you begin these future studies?
Yeah, so we've talked about that, and the protocol is submitted to the FDA for the EAP. Basically suggests there are no requirements for the EAP itself. You're supposed to monitor safety. And
right.
So what we propose is it's just the normal clinic visits, so that every 6 months is often what a child goes to see the clinic. And so it would just be that during those normal clinic visits that the physicians would report back if there's any new safety concerns.
Right, and so for those of the extended access, I'm familiar with a couple people who had to alter their dose. The ones you mentioned, that a few had some weight problems from at 6 and went back down to 2. So whatever dose you're on currently is the dose you'll continue on in the EAP. Is that correct?
But you have flexibility both in the LT and the EAP to adjust the dose. It's up to the physician and the patient, the family. So anything between 2 to 6 is recommended.
Okay. All right, thank you. And you said there was a middle dose of 4. That's not going to be in the study, but how do you figure out who needs to be on 4 or should change? Is that by your biomarkers that you're looking at, or your proteins, or is it by side effects that you might see in a given individual?
Yeah, so far the only side effect that seems to come up is the weight gain, as you mentioned, and it's in a subset of kids. So it's really up to the physician and the family to achieve that balance of efficacy and side effects. So we don't provide any micromanagement of that. And we're not measuring biomarkers or reporting those back.
Okay. And then in this current study, in the EAP, you're measuring you're doing irregular spine films like every 6 months to make sure that there aren't any accompanying compression fractures. You shouldn't have them based on the fact that Vamorolone doesn't impact bone metabolism or negatively impact bone metabolism. I think that, but just to be sure, are you looking at some of those factors?
We're doing spine x-rays at the last visit of both the LTE, the 2.5 years of treatment, so the kids coming off of that, actually starting this month, will be getting spine x-rays. Now the issue is, what do we compare them to? And so what we've arranged is a collaboration with the 4DMD study that Birch Griggs and Pete Bushby and Michaela. And so now we've, with some initial pilot funding from Foundation Eradicate Duchenne, we'll centralize all the films for 4DMD up with Leanne Ward, and for both Vamorolone and for deflazacort and for prednisone in the different dosing groups, and all be directly compared, which is a nice study going forward. Now in the EAP, so the Extended Access Plan, again, we don't require any outcomes other than just monitoring safety. So it defaults to standard of care. And so in that standard of care, as you know, there's increasing concern about bone morbidities, but it is up to the physician to decide what tests to do and when.
Right. So if it's just as an example I'm throwing out here, if we would see a child in the EAP that has some compression fractures, there's no worry that they couldn't do a bone sparing type intervention with them, right?
Right.
Okay. And so one of the other questions that has come up is the CK, and you mentioned how the CK drops with the use of Vamorolone. Do you have any data on whether or if it drops with deflazacort or prednisone?
I think I'm trying to remember, and don't quote me, but I think there's a bit published that actually goes up, if I remember right.
Yeah,
but
I can't recall the paper, Eric. I remember a paper on it, but I don't remember the data.
We're both trying to remember. I thought it went up, but we can go back and try to look.
Okay. I think that would be important information to really understand, or at least to have for the families that are interested. One mom contacted me some time back and said that her clinic said that her son's mutation didn't fit into the Vamorolone study, and I was confused because it's mutation agnostic. So is there any circumstance where a child would be excluded based on a mutation?
Well, the thing is you don't want Duchenne patients making dystrophin where they're really Becker patients. And so that gets tricky because, as you know, there's this reading frame rule. You can look at out of frame mutations and in frame, but it's not 100% predictive. So I think how the protocols are written is there's some sort of danger areas of the gene where you really start getting scared if you can predict Duchenne versus Becker, and that's mostly in the 5' end, the very beginning of the gene, where you can have out-of-frame mutations and still be Becker, making lots of dystrophin. And, I think it's there that we start getting worried. So that might have been the case. And what you can do in those cases is just show that the muscle is making no dystrophin. But, a lot of kids don't have biopsies anymore, and that becomes a problem.
Right. Well, I think that the biopsy issue is something that we as a community need to talk about again. Stan Nelson recently discussed at our clinical care summit just last week that biopsies are useful at certain times, necessary, but that we might kind of think back that a biopsy would be very useful to people looking at this disease. But that's a case for a different day. Eric, a couple other questions about Vamorolone. One of the fathers in this study on the EAP contacted me and said sometimes, and I think this is true of all studies, sometimes they hear of changes on social media as opposed to inside the clinic. So I think that if, and this is probably for you, Suzanne, as well, if there could be some sort of way to contact all the families should you make a change, whether that's dose, whether that's timing, whether it's visits, whatever that has to do with, that the patients could be notified by their clinical sites more rapidly. And as I say, this is hard because of the time difference in the UK. If folks are notified there, it's going to be on social media. So I'm not sure how we get around that, but I just raise it as an issue that is concerning not just your study, but every study that's happened about timing and who notifies the families and when and how that happens. So I just bring that up as well. So we're really excited about Vamorolone, Eric and Suzanne. And I guess if we think about the gene therapy studies that are also in early days with 3 companies and more sites over time, one of the questions is Vamorolone a sufficient immune suppressant to be considered, once it's approved, equal to in terms of use in a gene therapy study?
Sure. It's important to point out in both any dystrophin replacement strategy, whether it's exon skipping or gene therapy, it's never normal dystrophin. It's Becker-like dystrophin or some variant. And Becker patients still show inflammation in their muscle. And so it's broadly felt that any gene therapy or exon skipping would still be done in combination with an anti-inflammatory. And that's why most of the trials are done as an add-on to glucocorticoids. So towards that end, if Vamorolone is approved, it would likely replace the use of prednisone and deflazacort. And so in that case, it would be an add-on then to the exon skipping and gene therapy trials.
Right, and then you would expect in a gene therapy trial that in most of the studies, or at least the studies I'm aware of, whether the child is on whatever dose of steroids, weekend, daily, whatever, then as they enter the trial and gene therapy is delivered, they either increase the steroids a day or so before and keep them increased for a period of time during and after, just to make sure that there's significant immune suppression to the virus. So you would expect the same for Vamorolone. Vamorolone would become standard of care, and then they might add on in a gene therapy study a dose of another steroid to further suppress the immune system and then take it away once they're comfortable. Is that how you would see that?
Right.
Okay. And Suzanne, a couple questions for you. So my first question is, are you available 24/7? How do parents contact you through the study? And this is specifically for travel, or if there's a question about the study, are you a contact person so they could come and say, I have this question or this problem, and can you help me sort it out?
So I am pretty much available 24/7. I've been called at very odd times because of the time differences, and so that's fine. So yeah, I mean, I can be notified. I've called on vacation. It's fine. I'm glad to help. And I do not always have the answer, but I can always find someone. There was an issue, I think, with a sick child over a weekend. It might have been a holiday weekend. I didn't have the answer, but I could contact and get in touch with people who could give the answer that the parent needed. So that's how we try to work it out.
Okay, thank you. I also have another question for you in terms of timing for reimbursement. If I call you and say I've spent $50 and I need that reimbursed, how long does it take to get it reimbursed?
So within a month, but that's that's a long time. Typically, I try to get them in and out. It's been over the holiday and that kind of time period. We've had a little bit of a delay, but nothing over a month. One of the stumbling blocks is when I get a bunch of reimbursements for over a long period of time, and I've had a few of them recently. So I might have reimbursements all the way back from the beginning of 2A. So that's just really time consuming. But other than that, typically they get punched out right away. And listen, I have had people who just call me up and say, look, I hate to say this, but I really need this. And I move them up to the top. I mean, in the end, we're here to make it as least burdensome as possible. So if I have gotten calls and we try to work it out.
Great. Thank you very much. And I think in one of the slides you had a timing for approval. So if all goes well, you are submitting an NDA for approval in 2020, and then we would hope to see an approval by the end of 2020. Is that your timeline?
Or 2021. Yeah, it depends how long. It takes 6 to 8 months in orphan. We're going to try to get accelerated or breakthrough designation, which might speed it up a bit, but we were denied earlier this year. We're going to try again this year probably.
Okay. All right. Thank you. Well, I really so appreciate this webinar to both Eric and Suzanne. This has been really great and informative, and I look forward to seeing these next studies open and certainly progress toward steroid replacement therapy. I guess one last question for you, Eric, in terms of the daily dose of Vamorolone. Have you considered combinations, do you think, in terms of suppression? Are you looking at animal data to see if daily Vamorolone plus, a 1 time a week dose of a steroid would be useful in terms of suppression of the inflammatory pathways, or have you not looked at that yet?
We've tried one attempt at that in mice of doing different combination therapies, but it's really hard to extrapolate from the mouse studies with one drug, much less a bunch in combination. So no, we haven't thought of that in humans yet.
Okay. And this last question for you, Eric, and I think that the answer is no, but do you have any indication of pricing for those who aren't accepted in the study? Because the drug is not approved, I don't think you can sell the drug to anyone. Is that accurate?
Right. It's not being sold. We're in an extended access plan. You can try to recoup some of your costs, and so we've been working with foundations, and like Foundation Eradicate Duchenne has provided $100,000 this year to help fund the EAP access to the people currently taking it. But no, there's no charge to the families, and it has to be approved for that.
Okay, for those, so once approved, you're hoping to approve it in all of the countries you're studying. Is that accurate? And if so, is there any way for others? So I guess my bottom line is, are you considering compassionate use at any time soon? And how would others access if this isn't approved in their particular country? Would this be available in any other vehicle?
Yeah, that's hard to say at this point.
Okay, I suspect it will be. So those are questions that you can put on your plate and we'll come back to over time.
Next year.
So, all right. Anyway, thank you both for joining us on this webinar. I hope those of you on the phone felt it useful and informative. Please let us know if you have additional questions, and we will certainly circulate those back to Suzanne and Eric. Thank you all for joining us, and I wish you well. And this will be posted online as well within about 24 to 48 hours. Thank you all for joining us, and we'll see you again.
Thanks. Bye.